I come from a background of child psychology, so I taught kids with autism for about 3 years, and I really enjoyed it, and I had this really deep interest in science and ecology in general, particularly aquatic ecology. But it was a little bit harder for me, so I decided to go back to school, take a few ecology classes. I applied to the University of Michigan School of Natural Resources and Environment. I had heard good things about their program. I had been in contact with Sara Adlerstein and Jim Diana and Ed Rutherford to just talk about the program and what sort of projects were possible. I started working with Sara, and she also had an interest in mussels. Once we defined our research, I decided to come to the University of Michigan Museum of Zoology to learn how to identify species and learn a little bit about mussels and the different species in general. Freshwater mussels are extremely important to the aquatic community for many reasons. Not only are they filter feeders in the system, they also act as prey for other organisms. But especially important is they are indicators of biological integrity. They're extremely connected with the community because their larvae require a fish host, and they're also extremely sensitive to stressors. Problem is mussels are also extremely endangered, particularly in North America, where they're one of the most imperiled groups. And in Michigan alone, over 40% of the 45 species are state listed as endangered, threatened, or of special concern. Since habitat loss is the main driver in mussel declines, I was interested in looking at both local and landscape level habitat variables and how they were related to mussel distributions. The first thing I did was look at Michigan's Lower Peninsula and the mussel distributions there and how they related to large landscape level habitat variables like hydrology and temperature. And it seemed that gradient was the most important landscape level habitat variable for 9 of the species that I analyzed. So with these results in mind, I next wanted to look at how mussel distributions were related to local habitat variables like substrate size. So to do this, I spent the summer of 2009 in the Paw Paw River, which is located in southwestern Michigan, and I did mussel and habitat surveys there with the help of the Nature Conservancy and the Michigan Natural Features Inventory. Before I did those, I needed to learn how to identify the Michigan mussel species, and Renee Sherman, who is a SNRE U of M alum, was very helpful with that. To do the surveys, we measured out a 128 meter square plot, and using glass bottom buckets, we counted the mussels and identified them to species. And we found, similar to the large-scale analysis, that there were species-specific distributions. One species, for example, the ellipse, preferred very large substrate, about pebble-sized, and could tolerate a slightly unstable flow, as opposed to the three-ridge, which was found in smaller sized particles with flows that were more stable. So the goal of the second piece of our study was to compare larval sea lamprey distributions and mussel distributions in the Paw Paw River to determine if refinement to the lampricide applications was possible in order to protect those mussels and have continued success with sea lamprey control. And sea lamprey are invasive to the Great Lakes, and they have highly impacted the ecology here. In order to control sea lamprey, a chemical, a lampricide, is used in the tributaries of the Great Lakes. This chemical is meant to be selectively toxic to sea lamprey. However, some studies indicate that there are sublethal effects to mussels and other non-target species. Mussels, in particular, are narcotized for up to 14 days and left susceptible to predation, beaching, and displacement by the currents. To compare the distributions, I did a computer-intensive statistical analysis, and we found that there was a minimal overlap in the distributions, and the habitat requirements of larval sea lamprey and mussels were very different. Mussels were found only in the main stem of the Paw Paw River, where sea lamprey larvae were found in very low densities, if at all. And sea lamprey larvae were found in highest densities in the tributaries. The main stem of the Paw Paw River is treated as one unit. By segmenting the main stem into 3 units, we can avoid those areas that are important to mussels and continue treating areas like the lower main stem, where lamprey densities are high and mussels are absent. This way we continue to have success in the sea lamprey control, but we protect those areas really important to mussels. So I'm graduating from SNRE this year, and I just got a job with the Division of Wildlife in Colorado, so I'll be moving to Denver, working in their aquatic nuisance species program.